Hello, everybody. Melissa will be here shortly, and the presentation will start in probably about 5 minutes or so.
Michaela is the spouse of Joe, who's the president. And so she was visiting for me. And but it was the new wildland fire from the Grand Forest into the wildland certification course on my students. So right by the entrance, when you walk in, you know, there was a sign for midnight of 2016. Actually, let's just do that one. It went fine. Never decided. Today it got out of its line twice. It was a training class. That's good. Yeah. I didn't know what to do with it. But usually mine don't get outside the line. It's right outside the line.
Yeah, it just keeps ramping up.
You run away in the summer? Okay. But you don't have like a musty
Yeah. I'm just gonna have that as
You know, I said that in April or March. It's gonna be June before I came down. Yeah. Yeah.
You have one that's 5 and 1:30 today.
1:30 today?
Yes. Holy cow, yeah. Well, it's expected, that's not expectable to be
So Marty, what is the way to set up for the, so it's broadcasting now? Okay. I just want to see, oopsie, I did not mean to do that. Okay. And then is there, oh, there's 5 viewers. Nice. Okay, so can you see this? Oh. Okay. Okay. Hi, folks. We'll get started soon. We're just waiting for some last-minute folks to show up. In the meantime, you can admire these lovely rhododendrons that Marty brought me from the Pacific Northwest. Okay, yeah, we'll start soon. And I'll go back. Let's see. Oh, will it go back to the screenshot, or is it just gonna be the wall? Oh, okay. Okay. Okay, so then we'll. Okay. Soon.
Right, supposed to stand here. All right, everybody. I think we'll go ahead and get started. So thank you for coming to this special seminar. And of course, this is for Melissa Whitman's PhD dissertation defense. And I'll just talk a little bit about Melissa. So she hails from the Pacific Northwest, as probably most of you know, and she did her undergraduate Bachelor of the Arts in Interdisciplinary Ecology and Geography at Western Washington University. And she's been here on an NSF Graduate Research Fellowship in my lab. And I think you all know that Melissa has a penchant for the unusual. And that's pretty much kind of defined in many ways her career trajectory. So she's done a lot of research did a lot of research as an undergrad in unusual places. So Tasmania, Madagascar, these are islands, so she loves islands as well. And as well in Puerto Rico. So the work she did in Puerto Rico as an undergraduate involved an unusual taxonomic group of plants, the orchids. So another dimension of unusualness. And that work resulted in a very nice ecology paper that she published a few years ago. And so for today, for her dissertation work, she's worked on a different unusual group of plants, the rhododendrons, on another island in Borneo, in an unusual geographic location, Southeast Asia. So we're combining all of these things, and on an unusual soil type, so serpentine soil, which she's going to tell you about. So this is the topic of her dissertation, and again, thank you for coming, and I will hand it over to Melissa.
Get this, and then can you set it to? Okay.
Yeah, you don't want to. How about some intensity?
There we go. Okay. So I'm going to start off remaining calm. Okay, so now I'm actually going to start with the main kind of presentation discussion of my dissertation and research. This is a photo from the summit of Mount Kinabalu, one of the interesting places I've had the pleasure of viewing and having a stronger appreciation for the flora in some of these unusual places. So I'm going to start off today asking you to think big. So think about, are there rules in ecology? Are there kind of universal things that apply across taxonomic groups, across spatial scales, across different kinds of habitat, different ecological contexts? And what are the possible exceptions to this? Are there explanations behind exceptions? And this is kind of the overarching theme of my dissertation, is thinking about, again, are there universal rules that apply in ecology, and testing some of these. So I'm going to start off talking about one of the most commonly observed ones in ecology, and that is the latitudinal gradient in terms of species richness, and the higher amounts of basically species richness in the tropics. And that's something that has been observed across taxonomic groups. You have plants, you have animals, and these are different maps showing this. Again, you generally have the greatest richness in those parts of the globe. That can be related to things like the amount of energy, history of kind of glaciation events, history of all sorts of different things. But it's, again, this large-scale gradient. At more of a regional scale, local scale, this trend has also been observed in terms of elevation gradients. However, this is also something where there are exceptions that emerge. in general, there are, yes, many species at the lower elevation areas. Again, this is often tied with resource availability, but you will also have peaks in richness at very different areas. And that leads to questions of why. Rappaport's rule is a particular rule that I'm especially interested in. And it has a relationship that's very similar to patterns of richness. It's almost like the inverse of richness trends, where let's say you're at a low elevation, you have the highest richness, and it then declines. Rappaport's rule is the opposite trend in that you have the range size is going to be smallest for species at low elevations and increasing as you go up in elevation. Same is true for latitudinal gradients. There's been also exploration as to whether or not it applies to sea depth. But this is work that was especially made notable by Stevens in the early 90s, and is one of the things that I'd like to test. So another thing you can think of as kind of broad trends along gradients is also the characteristics of species change. And often that's a reflection of changing dynamics with other species, so levels of biotic competition, as well as the influence of things like seasonality, where when you're within tropical areas, you have reduced seasonality effects, whereas when you go to more closer to the polar regions, that's much more of a strong influence. Another thing you can look at is along an elevation gradient. This is kind of a more universal observation. You generally have much larger biomass, taller forests, at low elevations, and that as you go up the mountain, you'll see that the total biomass begins reducing, becoming less. There have also been a lot of natural history observations that things as subtle as leaf size also changes, where at low elevations you can have a wide range of different sizes of leaves, especially very large leaves. But then as you go up in elevation, everything kind of begins getting smaller. Things like the amount of intricacy, like palmate leaves, instead become more elliptical. That's kind of a general trend that's observed many different locations on Earth. One thing to also think about is traits and kind of energy usage or resource usage, and the idea that basically all life requires energy and resources in some way. However, there are multiple different ways in which those resources can be used. And you can think of this as a spectrum of different possible strategies. So you have, at one end, you can think of it as the slow and steady approach. And at the other end, the live fast, die young approach. And this is, again, for how resources are used. So do you go for leaves that essentially can last a really long time but cost quite a bit? Or do you go for ones that are more, well, disposable, that can easily grow? It's okay if they're eaten, because you can keep producing more. Again, it's this kind of, think of it as a spectrum of what are the costs involved. And that again is, everything has a cost. It's how you decide to budget. So here, I'm going to talk about, these are the three main components of my dissertation: and examination of the rules, and especially thinking about what are the exceptions to the rules and why. Is there an explanation behind this? So I'm going to start you off, again, examination of Rappaport's rule, and I will talk a little bit more about how that's calculated in a bit. But that applies at the macro scale. And macro scale in this case will be a large section of Southeast Asia. And then I'm going to focus in more at the regional scale to a single mountain and have comparisons between essentially the influence of things at the local scale. So that would be things like vegetation zones and soil associations. And so for the first two, that's very much focused on range size, richness trends. Whereas the third chapter, I make a shift and instead focus on the traits. And in that particular group, focusing on rhododendrons, one of my favorite genera out there. And talking also about ecological strategy. So thinking again about how resources are used, how species survive, what is their approach along a gradient. Okay, so starting up, we're going to have, again, this is on taxonomic variation. So I'm going to do a comparison now between 54 different plant families and the applicability of Rappaport's rule. And here are my questions. Essentially, is there a variation in land plants, specifically families and their strength of Rappaport's rule. Is it universal or not? Is rule the right term to use? And then, if so, is there any sort of variation related to the elevation where richness peaks? And that's at the family level. Elevation breadth, again, this is at the family level. How much of an area does that particular family occupy? Or growth form. And I'll get into nuances of how I define these things soon. Okay, so I'm going to start off. This is the area that I'm looking at when I say macro scale. So I utilized GBIF. These are kind of global herbarium record databases. And started off with over 1 million records. And then through a series of criteria, cleaning processes, narrowed it down to essentially 54 plant families that were especially species rich. And then of that, those represent over 15,000 different species that were used in the analyses. Some of the criteria included each species had to have a minimum of 2 points of data from different sources for elevation for where they occur. So this is not the full richness of the region, but you can think of it as a good representation, and also again with the most information available. Upper left-hand corner there is kind of a map showing the distribution of herbarium records. Okay, so now this is the nitty-gritty of how Rappaport's rule is calculated. So one of the first steps is, this is using Stevens' method, as it's referred to. You essentially take your elevation gradient or latitudinal gradient and divide it up into bins. In this instance, I have 100-meter increments. And then it's making the assumption that a species can occur at any point between the minimum and the maximum observed record. So that's what the red dots are, the min and the max. They can occur anywhere between there. And then for every single bin, you assess what is the richness. So for instance, in this first bin here, there are 3 species, but 5 observations. And then what you do is that for every species that is present, you calculate the extent or range size, which is the maximum minus the minimum, and then you average it for the species that are present. And Rappaport's rule specifically has the expectation that you have a positive slope that is produced by the average range size increasing with elevation. So this is kind of an example of what that would look like, where let's say you have this intersection point at the lower end. The average would be between these 3 species with a very small extent, whereas at the other end here, it'd be the average of 2 species with a very broad extent. And it's looking at that trend, again, of what the average range size is across these elevation bins. So that's how the nitty-gritty of at least the calculations. Okay, so now this is an example of differences between taxonomic groups, so kind of the first stage of really looking at the data. In this instance, I've broken it into 4 categories: angiosperms, gymnosperms, mosses, and ferns. And in pink, those are the richness curves. And in turquoise are the range size trends. So from looking at this, first observations, angiosperms, their richness is highest at low elevations. That's a kind of classic idea, again, that that's where the highest point of richness is. But for all other groups, their points of highest richness are actually higher up in elevation. So that's already an interesting point of contrast. Also, if you kind of look at it and squint, in the general sense, they all do have support for Rappaport's rule. But then as you look a little bit closer, you'll notice greater variation, especially at high elevation areas. And in some instances, there may actually be a change in the slope. So why is that? That's part of what I get to explore next. So again, this is the first broad look. Now, I'm going to take a closer look. And again, this is the replication across 54 plant families. And here is examples of the different gradient of types of things that you see. So, and whether or not there's strong support or no support for Rappaport's rule. And I refer to this as the strength of Rappaport's rule, specifically based off of the slope of the relationship between elevation and range size. So here at the no support end of things, you have rosaceae. They actually have a negative slope, which is completely counter. Or one of my favorite groups, ericaceae, completely flat. So those are definitely exceptions to Rappaport's rule. But as you get here to some of these other groups, so tree ferns, palms, dipterocarps, they have a strong support for Rappaport's rule that would match predictions. So already there's some differences between these families that's emerging. If you look up here, this is a histogram of the different slopes and the distribution. You can see, yeah, there are some exceptions, but overall, majority of the slopes are positive. And most of these, again, do support Rappaport's rule. So now I'm going to get into, okay, yeah, there's differences, but is there a common explanation for when it does and does not apply? And for this, I look at trends across plant families. So for this, the important thing here is the response is, again, the strength of Rappaport's rule, that slope metric from 54 different analyses. So that is, so again, this is the trend across all plant families. Can it be explained by things like the differences in peaks in richness? 1 second, let me grab some water. So first to observe, majority of the species, or majority of the families rather, they have their peaks in richness at low elevations. But then other groups, such as orchids, their peak in richness is going to be more mid-elevation, associated with things like cloud forests. Then up higher still, this is where you have, this is a moss and ericaceae, so those are more upper elevation. That's their peak in richness. And then you have extreme outliers, like rosaceae, which they're really only found at the highest places. So that's kind of interesting to begin with, like, what is the distribution of these different families? And this is an example here, nutmeg. Again, their peak of richness is going to be very close to sea level. Here, orchids, their peak in richness, moving up. So you can get an idea of how this relationship is. That is the y-axis. Does that define, again, the location of their richness peak? Does that explain differences in Rappaport's rule? The answer is yes. And I'll get into ideas as to what exactly species richness is indicative of. Also, I examined distribution breadth. This is a different metric, and this is at the family level. So for instance, here, these are dipterocarp. They are extremely restricted to only the lowest elevation tropical wet forest. The distribution breadth for the family as a whole is only about 1,000 meters. Whereas you have groups like ericaceae here, they are found all the way from sea level to mountain summits. Overall, for that family, they have a very wide distribution breadth. That actually becomes an even stronger predictor of the strength of Rappaport's rule. And what's nice about this is you don't have quite the same clustering as you did before. It's, again, disentangling when and when does it not Rappaport's rule apply. And I also examined growth form. And this is a weak significance, but it's still significant. And that is arborescent. So tree-like growths, they have stronger support, whereas herbaceous groups, families that are dominated by that growth form, really don't. And so that begins kind of also raising new questions, again, about what type of things influence. You can think of it as the parameters of Rappaport's rule. What defines it for when it applies, when it does not. So this is a quick review of findings. So again, the strength of Rappaport's rule varies between families. And Rappaport's rule applies when a family either has richness peaks at lower elevations, a very narrow distribution breadth, or tree-like growth forms. And so now I'm going to get into the broader interpretation of why richness peaks, why elevation breadth, why growth form. So again, there could be, these are parameters you can think of that help define the rule. And You can think of potentially peaks in richness as being an indicator of things like the biogeographic history or evolutionary history of each family, thinking of them as potential points of origin or initial colonization points. You can think of distribution breadth as indication of the tolerance of a family to different kinds of habitat types, the amount of time frame that they've been in an area, being if they've been in a location longer, broader. And growth form may be a proxy for ecophysiological limits. So for instance, tree form, they're going to have a lot different types of limits on where they can grow based off of resources, the size, just the movement of things like water through their systems compared to an herbaceous plant, which is very small. So potentially they have additional limits. So you can think of the combination of these things as being kind of the factors that influence range size, the distribution of species across the landscape. So now I'm going to move to the next section, which is, again, Rappaport's rule. And in this instance, we're going to shift to the regional scale. And one of the key things is that I'm going to be taking a look at vegetation zones and soil associations. And the big serpent here is because I'm interested in ultramafic soil, which is also commonly referred to as serpentine soil. So my questions here, does Rappaport's rule apply at the regional scale? One of the things with a lot of these broad scale rules is, again, does it apply at multiple spatial scales? And often things that are noticeable at the very broad sense, as you get to a smaller, more local scale, it begins to kind of not apply as well. So does it still work at the regional? And that do range size trends, are they a reflection also of vegetation zone boundaries or differences in soil association? So here we're going to take a look at ye olde historical picture. Von Humboldt. He had a number of expeditions to notable places of the globe and observations of things like vegetation zone boundaries. And here, for instance, lowest elevation, region of palms. And then as you go up, region of arborescent ferns. And then taking note of things like, where is the tree line? Where do you cease to have tall woody growth and you're restricted to only shrubs and grasses? And one of the key things is not only does the vegetation communities change, but also what factors might be causing that. One of the key things is position of the cloud layer. So that is generally more mid-elevations. Within the cloud layer, you have more like a mist-like immersion, whereas below, you have more like rain. And above, you can actually have more of a drought type of conditions because, again, the moisture's below you and there's more infrequent moisture availability. Temperature also declines in a very linear fashion. So you have two different strong factors that change along an elevation gradient. One of the key things I should note is that elevation gradients are wonderful to study compared to latitudinal gradients just because you have so much more compressed into a smaller area. And you can, observe multiple different habitat types within the same given region. Okay, another ye olde historic researcher is Robert Whitaker. He had observations in the Siskiyou Mountains, especially, of distinct vegetation communities associated with ultramafic soil. So ultramafic soil is interesting in that if you're at a given point in elevation, there's dramatic differences in the overall biomass available. So for instance, normal other types of soil, you have for full-size trees, other types of vegetation, whereas as soon as you get to the ultramafic, the most kind of first impression is everything is much shorter, smaller, and more distantly spaced apart. There's more light, but the thing is that there's not very much in the way of nutrients. There's a combination of either extremely low nutrients or imbalances in the nutrient availability that make it more difficult for many plants to grow. One of the key things is often there's heavy metals and high amounts of toxicity. So many plants that grow on this type of soil, many of them are endemic or specialize also in that soil type because of extremely high amounts of things like nickel and cadmium that most other things just simply cannot tolerate. There's also reduced water holding capacity. And one of the things about it is that these patches of soil are in many ways like little islands on the landscape. And Whitaker's observation of this helped shape ideas of our understanding of not only regional richness, but also kind of what is the turnover in species richness between sites, or beta diversity. And so these are two different kind of concepts of thinking about how our vegetation community is shaped, both along an elevation gradient in terms of things like temperature, climate, and in terms of also edaphic conditions. So this is a photo here, Mount Kinabalu, exact same elevation, two completely different canopy heights and amounts of biomass between ultramafic and non. So again, I'm shifting from this entire big macro scale to just this area in the northern part of Borneo. So Mount Kinabalu. And the wonderful thing about Mount Kinabalu, it's an extremely distinct mountain for the region. And there's a huge amount of natural history information for that site. So with this, I tapped into a different set of information. So making use of actually multi-generations worth of records. So utilizing the Mount Kinabalu herbarium database, and that's in partnership with Reed Beeman. His father spent his lifetime collecting plants there, as well as there's been an entire, you can think of it as over a century worth of collecting at this site because it is such a notable mountain. So you have a great deal of local information. And one of the nice things about these records is it included observations of soil conditions. And again, because ultramafic soil is so distinct, people are able to write about that within the observations of the plants. Usually soil associations require more detailed testing. But here I was able to combine all of this together and take a look at this and narrow it down to around 3,000 species from 193 families. And again, this had additional criteria for more information availability. So you had to have multiple observations for every single species, as well as information on some of the soil specialization. And that, yeah, I'd also tapped into additional books and literature. Here's an example, for instance, of a rhododendron from an ultramafic site that's been dried in the herbarium. I got a chance to visit that. And you can see the white on the leaves. That's actually excess minerals that have accumulated on the outside when it's dried. So that's in response to extreme imbalances between calcium and magnesium, for instance. And I also looked at the literature for the area. And there's a lot of observations of things like distinct vegetation zones as you climb up the mountain. And Kitayama referred to his work, noted distinct breakpoints at 1,200 meters and at 2,800 meters, essentially defining low elevation, tropical rainforest, the area of cloud immersion, so the cloud forest, montane areas, and then the area above, the subalpine. And so these are big groups, but even within a given area, you can also have distinctions and more subtle variations in vegetation zones. And so for statistical methods, I broke the data set by soil association and replicated all analyses based off of soil association. And I used, you can think of it as the most common approach for studying Rappaport's rule, which is linear regression. Range size as a function of elevation. Very simple. As compared to two other model types, which is range size based off of these observed or forced breakpoints. So that's piecewise regression. And then the third one being also piecewise regression, but essentially letting the data decide where should the breakpoints be. So that's allowing flexibility for, let's say, is there potentially more vegetation zones than what I am forcing, or are they at a different location than I had anticipated? And then using AIC, Kaiki Information Criterion, to decide what is the best model selection. And then in addition to that, creation of a series of null models. And for the null models, that is really testing, does soil matter? And for that, that's a random shuffling of soil associations, but keeping things about what are the min and max for where species occur, that is kept constant. So this is the best models for each of the soil groups. And so you have ultramafic in orange, What I refer to as tolerant, those are species that are not specialized, not endemic. They're found some of the time on ultramafic soil, some of the time not. Often, the tolerant species can also be more early successional, so they can go to kind of opportunistically whatever is available. And then blue being all other species. And so they can also have specialized soil associations, but it's not ultramafic. That's the key thing. And across all groups, piecewise regression was always better than linear regression. So even though piecewise regression is more complex, it better matches the data. So that is what was selected using AIC selection criterion. And that in itself is a distinction compared to most studies on Rappaport's rule, which again, look at the entire gradient at once, not subsections. And then the other thing that was interesting is that differences in the breakpoint locations. So if you look at the non-ultramafic flora in blue, their breakpoint locations actually were very, very close to those observed in the natural history literature, which makes sense because this actually also represents the vast majority of the species for the area. So that reaffirms that yes, vegetation zones matter for determinants of range size. But what's interesting is that for the non-ultramafic flora, there are differences in the breakpoints. There's some similarity at the transition, for instance, between low tropical forest to cloud forest. But one of the things that is also more interesting is this point right here, where they have a distinct trend. This is the point that actually corresponds with where the tree line is, where there's freezing events. So that's a strong determinant of essentially which species can and cannot handle that kind of stress. That's more of a hard boundary, and why there's a distinct change there. In the instance of the non-ultramafic flora, at the transition at the subalpine zone, so a little bit farther down, that's more of the break-off point for them. Either they can handle it or not. So that's why you have a different trend here. Overall, you see the greatest transition at the mid-elevation areas in the slope range. So that's the steepest slope. So now I'm going to talk a little bit about the null models. And again, the null model was created via randomly shuffling soil association, but keeping things like richness constant. So if I had, let's say, 50 species ultramafic, I would still have 50 species in the null model, but the classification, or more the minimum and maximum values would be randomized from the data set that's pre-existing. So it would be a mix of non-ultramafic, tolerant, and ultramafic potentially. But again, richness would remain the same. So in these figures, anything that's solid is a significant difference from the null model. So this is essentially replicated for every single elevation bin in 100-meter increments for is there a significant difference from the null model or not. And one of the things you can observe is that the range size trends for ultramafic and and tolerant species are much larger. So that's a larger intercept. Also, the slope is actually fairly continuous within the mid-elevation areas. Whereas non-ultramafic species, they have a much smaller range size than anticipated. And there's much more dramatic kind of zigzag pattern. So they are more heavily influenced, you can think of it as, by vegetation zone boundaries. And I'll get into more interpretation of this in a moment. But these are the main observations from the null models. So quick summary here. There's overall support for Rappaport's rule if you kind of squint and look at the gradient as a whole. However, this begins changing as you begin incorporating more and more information. So that would be information on soil and information on vegetation zones. And when you view things via subsets, you actually begin losing support for Rappaport's rule. In many instances, it does not apply. The strongest support for where it does apply, though, tends to be mid-elevations. And that at the regional scale, models that include vegetation zone boundaries are better predictors of range size trends than elevation alone. So that's the piecewise model selection. And that specialization or tolerance of ultramafic soil creates distinct range size trends. So again, that is differences in the overall, they have larger range sizes and with less variation across the gradient. So now I'm going to talk a little bit about broader interpretation of why, what might be happening. So if you think of it, flora that specialize in extreme soil types, edaphic stress may actually be a stronger determinant of where they occur than climate. So whereas for species that don't have this kind of association with the extreme end of low nutrients, they're going to be more influenced by things like temperature and moisture availability, hence the vegetation zone boundaries. And then this is a different idea also, is that tolerance of one stress may now enable tolerance of other types of stressors. And there's also something that can emerge that potentially the much larger range sizes, that's this idea of the potential niche. So you can think of it as, let's say for an ultramafic species, they can occur across a very large elevation gradient, different kinds of conditions. But when you're actually going and doing field surveying, you may find that they are very, very restricted to just that soil type. So it's this kind of difference in the potential niche versus the realized. Essentially, areas beyond that soil patch, they have competitive exclusion. So these are some of the different ideas behind this chapter. And now I'm going to shift to the last part on rhododendrons. Okay, so now we get, again, the shifting of, rather than focusing on richness and range size, thinking about the traits that a species has and how that changes along a gradient, and thinking about how resources are used and how that also is a reflection of the ecological strategy for survival. So is it possible to maintain a single ecological strategy along the elevation gradient? Or does the strategy have to change as you have this transition from mostly biotic competition-based stressors at low elevations, where you have more plentiful resources, to instead a transition of more abiotic stressors as you go up in elevation? So does your strategy have to change with that gradient? And then how might species, specifically with a stress-tolerant ecological strategy, and you can think of that as, again, the amount of investment they have in their leaves. But one of the key things is also things that are height limited and very slow growing. So think of that spectrum again that I mentioned at the beginning. These would be flora at the super, super slow end of that spectrum. How can they exist within low elevation areas and avoid competition if they simply cannot grow faster, taller than their neighbors? So how might that work? So kind of a quick overview. I'm going to be talking a lot about functional traits. So quick definition. Oops. What are functional traits? Essentially, it's a standardized metric that's useful for comparisons across taxa. And most importantly, it's a strong indicator of how resources are used. So one of the major ones that's used is specific leaf area. And that's a metric that's based off a combination of leaf area as well as weight. So you can think of it as the amount of investment in a leaf. In general, plants with very low SLA have very thick, tough leaves that are very dense. A lot goes into them. They're highly protected, but it takes a lot to grow, and they're very, very slow. Whereas ones at the other end of the spectrum, you can think of them as the fast end. Nitrogen content, again, that's a direct indication of how our resources used, what kind of resources are available. And thinking about these traits, a lot of these do form pretty specific rules in that there's known trade-offs between traits. Strong correlations where essentially if you have low leaf nitrogen, you're going to have low SLA, for instance. So again, high resource availability at the other end is going to be more of the live fast, grow lots of leaves, but less investment type of approach. And you can also think of, again, leaf traits as an indication of habitat type. And here on the left is an example of temperature and moisture and how that shapes different kinds of habitat communities. And you can kind of think of this as a gradient of resource types, where at the highest resource availability, the most amount of energy. You have tropical rainforests. So those would be, again, massive biomass. In areas that are extremely limited, you have the other extreme, tundra, where, again, your biomass is itty bitty. And then you can also begin thinking of how do these habitats relate then to the leaf traits. So it's making this connection between a larger scale to an individual plant, individual leaf basis. So here, LMA, that's very similar to SLA. It's more like the inverse. And nitrogen mass. So again, this is the spectrum idea of how resources are allocated and how are they used and also what's available. Okay, so now I'm going to revisit Eric A.C. So remember in the very first chapter, they had no relationship with Rappaport's rule. They were an odd exception. And in chapter 2, ultramafic species, those were the odd exceptions. And within Eric A.C., there's this lovely genus, rhododendron, which also occurs on ultramafic soil, Southeast Asia. So it's a good kind of fusion of the exceptions of both other chapters. And the nice thing about rhododendrons is that they are an incredibly diverse group. So there's over 1,000 species worldwide. They occur from the tropics to the tundra. Within the tropics, they occur all the way from sea level to mountain summits. And so a huge variety of habitat types along that elevation gradient. They're generally regarded as stress tolerant. And yes, there are species that are specific to ultramafic soil. So that's going to be my model group to test. How can they occur across such a large gradient? Might it help explain why their range size remains constant also? So here, this is the theoretical kind of reference point of kind of what I had in mind. Where blue are trends that you observe across the entire gradient. This is across species. You're reading the literature. You're doing field surveys. You have a very high amount of, you can think of it as functional trait diversity. Many different possible ways of surviving. Different kinds of leaves, characteristics at the low elevations. And that as you go up in elevation, you have increasing amounts of abiotic stressors. And that kind of forces all of the plants into a kind of narrow, you have to be able to handle this stress. And if you can't, you're excluded. So this would be the expectation based off of the majority in blue. However, what I'm looking at is this question of, is it possible to maintain a single strategy across the entire gradient? And with the single strategy, from that you would expect actually very little amount of functional trait diversity. It's like you have one approach, that's your thing, that's what you do and you do it well. But it's restricted to that. And how would that look across the gradient? And specifically for stress-tolerant flora, I would expect them at the very low end of the gradient. So here you have the diversity of types of traits. And at this size, you can think of it as life history strategy. You have the very slow stress-tolerant at this end versus fast at the other end. And so expectation is that rhododendrons would look something like the yellow here. Okay, and so to answer this, I tapped into wonderful live plant collections around the globe. So the wonderful thing about rhododendrons, random trivia, third most popular plant after orchids and tulips for Victorian era naturalists to collect. And so you actually have these massive collections, entire greenhouses devoted just to that genus. So one of the places that I worked was at the Royal Botanical Gardens in Edinburgh. They have over 600 species in their live plant collections. The other thing that's really nice about rhododendrons is you can grow them from a single cutting or from seed very easily. And that means that most of the plants represented are all wild collected. So these are essentially serendipitous common gardens. Many of these plants, though, are very slow growing. And so the average age of the plant that I sampled was almost 3 decades old. So that's something that under the normal dissertation timeline would not be possible, but I made use of this. And I also sampled at Bovie's Rare Plant Nursery, which is one of the largest private collections, as well as the Rhododendron Species Foundation, which is the largest collection in North America. So 3 different gardens tapping into these wonderful history. Okay, so that gives you an idea of my desk setup over the summer. I sampled information on 169 different rhododendron species, and I had a number of different traits that are really specific to just the leaves. And for every individual plant, I generally collected 5 leaves. I had a very standardized way of measuring every single one. And then whenever possible, I tried to collect multiple individuals. All of this trait information was then averaged by the species. Quick note. I also made use of information from herbarium records and from botanical monographs, such as by George Argent. He was my collaborator in Edinburgh, who had information on what are the elevations that these species occur, and also making use of some of the other databases used in chapters 1 and 2 to fill in information gaps. And then this also provided information on habitat association and growth form details. So quick look. How do rhododendrons compare to other plants? So this is rhododendrons on the Global Plant Trait Network. And again, this is the spectrum. Here's the slow end of the spectrum, fast end of the spectrum. And this is log transformed because that's generally how it's represented. And you'll notice rhododendrons, they're, yes, at the slow end. So that confirms, yes, they're mostly stress tolerant. One of the things, too, you want to look at is the slope. Their slopes are parallel. So they follow the rules in that regard as far as known trade-offs. But they do differ in the intercept. Essentially, they can make do with a lot less, is what that's saying. Is that for any given amount of nitrogen, they can have a slightly higher SLA. Okay, so what other ways of comparing rhododendrons to other taxa? So rhododendrons are very similar to conifers. And to other plants, they're associated with heathlands and extreme low nutrient areas. So these are different major taxonomic groups. Rhododendrons would be around here. That's where, again, you have, this is a comparison to evergreen or needle-like species from the data set. That's kind of where they are. So they're actually going to be closer to conifers than to other flowering plants. And what's interesting is if you look at some of the tropical species, they've actually changed from these broad elliptical leaves to needle-like leaves. This is rhododendron ericoides. They have itty-bitty little needles. They look almost like a Douglas fir. And they're found at, for instance, the summit of Mount Kinabalu and on ultramafic soil. Okay, so how do these traits change along the gradient? Well, many of them don't. That's the key thing. You don't have a relationship between many of these different traits that are thought of as indications of life history strategy. So SLA, leaf dry matter content, thickness, toughness, carbon-nitrogen ratios, density, even multivariate metrics of functional diversity. There is no relationship with elevation. There is some degree of scatter, so there's going to be differences between species, but it's not associated with elevation. However, there are some traits that do correspond with elevation, one of which is leaf size. And that actually matches this paradigm from natural history observations, where low elevations, you have really large leaves, and that as you go up in elevation, they become small. So this does support then that they do respond to their environment, yet they keep their strategy relatively constant. Also, one thing of interest, nitrogen. That goes up in value with elevation. That is completely counter to most other plants, yet it has more parallels with plants that are found more in alpine areas. And that's in instances where you have things other than nutrients limiting the maximum size of the leaves. That can also occur, for instance, if you have plants that have full sun exposure. So the leaf size that's required for photosynthesis is very small. So again, it's things other than leaf nutrients defining leaf size. So this is an example. This is rhododendron ericoides at the summit of Mount Kinabalu. I climbed that. They have a lot of light up there. But it's also an extreme habitat in that you have huge amounts of temperature variation between day and night, huge amounts of wind, lots of solar radiation, all these other stressors happening. And this particular plant is so well adapted to low nutrients. It can handle ultramafic soil. It can handle granite. It's doing just fine. It's one of the only things that can handle that level of stress. And so now I also performed a meta-analysis. And rather than using the prior data set, I specifically looked at other tropical woody plants along an elevation gradient, looking for what are the two most common functional traits, SLA and percent nitrogen. And how do rhododendrons compare? And this helps give you an idea, really, for comparison of how are these plants along a gradient. And it kind of looks similar to my initial predictive little diagram, where you have rhododendrons in kind of this narrow band across the entire gradient. Again, they're at more the lower end of the spectrum in terms of their traits, compared to other plants, which can have a huge range of different leaf values, many different strategies. And again, this idea that as you go up in elevation, you begin having this narrowing of trait space. So again, rhododendrons, SLA, no relationship. And this is up at the top is nitrogen. Most other plants have a decline in nitrogen. Rhododendrons, slight positive. So how does this look at the extreme end of the stress spectrum, even within rhododendrons? So they're stress tolerant, but even within that realm, again, there's some areas that are even more stressful. So that would be being able to grow as an epiphyte or growing on ultramafic soil. And for this, I did more of a multivariate analysis, the total trait space. And guess what? There's no difference actually between terrestrial and epiphytic plants in terms of their leaf characteristics, and also no difference between other soil types, and again, this extreme ultramafic. The ultramafic, though, did have the distinction of individual traits. Some of them did have significant value differences. So SLA, for instance, was generally lower for ultramafic species, same thing with nitrogen. And this overall space was at the lower 15th percentile. But if you look at all the traits again, both the mean as well as the dispersion, the kind of the breadth, there's no difference from that perspective. Okay, so summary here. I'm going to try and wrap things up. Essentially, rhododendrons, yes, they're able to maintain a stress-tolerant strategy across the entire elevation gradient. And that expansion into lower elevation areas, again, this is where most of the time they would be excluded by competition. They kind of found their own way around it. And that this is potentially facilitated by making the transition from terrestrial growth forms to epiphytic. And just that transition alone is pretty amazing if you think of it going from a tree to something that can handle, well, growing on top of other trees. And that the specialization also in extreme edaphic conditions, that's an instance where if the soil is found at a lower elevation, they're still able to then avoid competition, biotic competition for things like space and light. One of the commonalities with all of these things is that they can handle low nutrients. And that's why, for instance, the distinction between terrestrial and epiphytic, there's not that much of a difference. If they're not used to having soil nutrients to begin with, it doesn't matter where they're growing. Same thing with ability to grow on things like granite. And one thing to note is that this genus also has very strong associations with things like mycorrhizal fungi. And so that's something where they're able to essentially get a little bit of help for the low nutrient areas. Okay, so what other kind of taxa are associated with nutrient poor habitats? What has become of them? So in the instance here, you have things like podocarps. They have become very restricted in their range, limited to essentially mountaintops in the tropics or ultramafic soil. Whereas other plants that are restricted to low nutrient areas, they've been able to expand. Some have global distribution. And one of the key things is innovations, such as parasitism, symbiosis with fungi, becoming epiphytes, or becoming carnivorous. So those are two different ways of responding if you want to keep your same strategy. You either, again, you stick to that, your range becomes restricted, or you have an innovation in a very different sort of way, and that allows you to new areas. And one thing that I like to think about is, okay, what enabled rhododendrons to do all of this? And for this, I refer to natural history information. It's thought that the genus originated actually in the foothills of the Himalayas and then extended down into the tropics, starting with mountains, and then expanded from mountains to lower elevations. So if you think of it from this directionality perspective, they're starting off from already being adapted to many stressful environments. They're used to seasonality, used to temperature extremes and low nutrients. They're starting off at the mountaintops, and for them, the main challenge then would be how to avoid biotic competition to expand into low elevation areas. So now this is the broader interpretation. A description for this, I would like to propose this steady stress novel niche hypothesis as a description of the strategy. And essentially, it's being able to maintain a stress-tolerant strategy, a narrow set of functional traits, but with that comes an expansion in a very different kind of axes of kind of characteristics of the plant. So that would be the transitions to epiphytic growth. It could be, for instance, tolerance with extreme kind of unusual events, so occupancy in disturbed sites, some other way that they can be very flexible to allow for opportunistic occurrence in different areas. And that this idea is directionally dependent. So again, it's going to be much easier if you have the point of origin in a stressful site already, high elevation areas, and expanding to low elevation, versus if you're low elevation and you don't have adaptations to those stressors going up, you're going to have to change. So there's a directionality component. And I'd like to test this idea further with other stress-tolerant flora. I think that'd be really interesting for the future. So now the closing thoughts kind of take home from dissertation as a whole. And that ecological rules underlying patterns of species richness, range size, functional trait trends along elevation gradients, those are far from universal. However, these exceptions provided very interesting insights into where and why these rules apply. What are the parameters of those rule spaces? Is there a single explanation or perhaps multiple explanations for why there are deviations? And that in general, the deviation from the rules, the rule breakers, were mostly associated with high elevation areas, extreme soil types, or flora with a unique ecological strategies or unique niches. So there you go. That's my dissertation. And so I'd like to thank everyone. I have so many people to thank. My acknowledgement section is 3 pages in length. So definitely I'd like to start off with my advisor. He managed to advise me and put up with me for all these years and all my quirks. And it's probably not the easiest thing, but it's been a good learning experience. And my committee, lots of very useful feedback over the years, very practical advice, also just general recommendations to stay calm, it'll be okay, and to think of the big picture. Also, lots of nice people, and hopefully some of you are also watching in remote corners of the globe because I'm live streaming this. So folks associated with Russo Lab, lots of wonderful feedback from you through all the different meetings over the years. Fellow grad students, the community as a whole, UNL faculty, staff, special thanks also for Kathleen for helping with the nutrient analyses, families, friends. Marty even brought me rhododendrons from the Pacific Northwest, that's in my hair, for support. Collaborators. I have many collaborators, but some of the main ones, so like Reed Beeman, he helped out with the Chapter 2 data set. Again, it's this kind of family legacy of Kinabalu data. George Argent with helping, especially with rhododendron, this long history of knowledge and access to the largest collection of rhododendrons on Earth, to the plants themselves and their lovely leaves. Funding, we have National Science Foundation, Graduate Research Fellowship Program that made a huge part possible. American Rhododendron Society provided me also with grants, SBS special funds. I also indirectly thank Juping. He introduced me to Bitcoin about a year ago. That's how I paid for my last semester here, because I bought a Bitcoin and sold it. So that final semester universal fellowship, I guess. Data sources, all the botanical gardens. So Edinburgh, Rhododendron Species Foundation, Bovies, Saba Parks, GBIF, all those folks. So there's a lot of folks. So thank you all. So there you go. Yeah. Yep.
Do you think these results will change if you actually consider the entire area?
I imagine they might. That metric, though, is really hard to get that level of detail for so many species. So it's kind of a trade-off. When you're doing big macro-ecological things, you kind of sometimes have to accept you don't have all the data you would like, and going for what is most available. That is something, though, at, let's say, a local scale, you would be able to potentially get that information, and where definitely area does become an influence. And if you look at things like species richness area relationships, that's another study for big trends. There's definitely a relationship there. So I imagine that would apply for other components too.
Could you make that estimate on the Mount Kinabalu data?
I
mean, just
possibly
assume it's a cone or something.
Yeah.
And I don't know how, it would be rough, but.
It'd be rough, and that might be possible because that is a more surveyed area. I think the challenge there is getting the high resolution data. That's something that I need to check into that in the future, but I know that there's been increasing interest of trying to get LiDAR data, for instance, from Mount Kinabalu, and that would be especially useful for doing area estimates and biomass estimates. I did do a very generalized area relationship for Borneo as a whole a while back as a preliminary one, and adjusting things like richness to the area available. And in that instance, rhododendrons are even more bizarre than other plants, and that their richness went up with elevation. And elevation, as you go up in elevation, you have lower and lower area. But actually that makes sense if you think about it, because in that instance, speciation is driven by these kind of islands of mountaintops, so to speak, and the separation between populations. And then I was like, oh, okay, that makes sense. It's counterintuitive. Any other general questions? It can be anything in particular. It doesn't have to be a dissertation.
What are the conservation concerns for rhododendrons in that area, especially with global change?
Rhododendron is a really interesting group. At one end, for the genus, you have things like Rhododendron ponticum, which are listed as invasive, horror of horrors if you ever go into Europe, and they talk about it taking over everything. But at the other end, you do have some species that are listed as critically endangered. There's actually international conservation red list for rhododendrons in a special publication that was produced. Many of the species, especially the ones that are really site-specific, so that would be to a single peak or single soil type, many of those are endangered. And that's where definitely I think having greater awareness of these species is good. One of the things that I like especially about the genus is that it's a tropical group, but it's one that's very well known. Like my mom knows about rhododendrons, my grandma knows about rhododendrons. It's one that it has a very strong horticultural appeal. So because of that, I think having this conservation messages is going to be easier than other plant groups because they are flamboyant and beautiful. One interesting thing as far as practical applications is that when I went to Borneo, there are some sites, there's actually a former gold mining site. And that is actually, I can, well, I guess the presentation's down, but that's the picture that had like this bright turquoise. That's actually the soil and the water conditions. It is so extreme there, but there are rhododendrons growing. There's like nothing else, but then you have these happy rhododendrons. And that's a really interesting example of where they're colonizing a completely new habitat type created by humans that is almost impossible for most other life. Yet rhododendrons are, again, they already have a lot of adaptations for extreme low nutrients, for high acidity in the soils. They're able to colonize in. So potentially rhododendrons themselves may also be a tool for phytoremediation, for habitat cleanup. So that's another appeal of the group.
saw interesting example of your stress-tolerant novel niche canoeing in Canada on a bay of Lake Huron that had been a logging operation a century ago, and they had filled this entire bay, which was probably 30 acres, with wood chips and sawdust. Just the entire thing.
The entire.
And as we approached it, it's like it was something strange. You could just tell them. It was the sun dunes. The entire, it was just a mat of carnivorous sun dunes. The carpet.
Yeah.
So that was a novel niche.
Exactly.
And that bog plant was just the right species for that nitrogen limited environment.
Yeah, I should actually, the mining site had the most expansive collections of nepenthes, the pitcher plants.
Yeah.
That's like the easiest place to see them. You go to the extra toxic sites, and there are these massive carpets of these beautiful carnivorous plants everywhere, and rhododendrons. And so I'm like, yay, all the quirky plants in one place. Yeah.
So what do you think is like the next biggest question that you're like really interested in? Like, what do you think from like all the stuff that maybe it doesn't have to be like the biggest, but like what's something that's really interesting to you like now based on what you've learned from like the business?
Wow, there's all sorts of different ones. I think what I would be interested in exploring is incorporating some of the phylogenetic information about rhododendrons. I think that's something that is on my list of I wish I had time to incorporate into my dissertation, but I want to finish categories. But I still want to do that. So I think having the phylogenetic information combined with the trait information, that can offer really, I think, stronger insights on rhododendrons along these gradients, what kind of trends are associated with particular clades versus location. So disentangling that more. Also, because rhododendrons are conveniently this global genera, taking a look at things on a latitudinal basis. My super preliminary look at it is that they also kind of break the rules in that regard. They have, for instance, the tropical species have much, much lower SLA. And as they go to temperate areas, the SLA goes up. So that means essentially the slowest growth, the most extreme is in the tropics, and it actually gets easier as you go to polar areas, which is, again, this very counterintuitive idea, but it's kind of interesting to think about why might that be. So that would be another on my list. And then I think it'd be interesting to get into the completely different realm of thinking about other groups of plants. So the things like orchids, I have a special spot in my heart for orchids, that do have these really complex relationships with other species, both for nutrient acquisition as well as for pollination.
Right. We'll wrap it up and we'll go get some lunch and then have Melissa's defense later.
Thank you all for attending.
And by the way, we're having a celebration.
A celebration or a decompression, one of the two.
My house, I think. Well, I'll write my
write the address up there.
And we're all welcome to come and stay for as long.